Bendy told you about his grandmother, but the truth is, all our grandmothers had COVID, and I mean, they are all fine, but that got us all interested into COVID, which is why we decided to work on this project, which is tackling disease and single cell genomics using machine learning with single cell. So, today we will talk about the aim of our project and how we got our database, how we clean up our data processing where we remove technical effects, and biomedical analysis. Firstly, let's go into the aims of our project. So our first aim is to compare different methods of quality control and single cell data analysis. And our second aim is to understand molecular mechanisms during severe COVID-19 infections in order to find the starting points for developing treatment. So the data we were working with were data from 6 patients, 3 of them healthy and 3 of them diseased with COVID. And each sample was 1,500 cells, which was in total 9,000 cells. So how we got our data was through RNA sequencing, which is a method where we reveal the presence and quantity of RNA in each sample. And how it works is we first prepare our tissue, so we have to separate each cell, and then we go on to single cell isolation, which I will explain later. And once we get our cells into droplets, we dissolve the droplets and put them into a sequencer, which then generates our data so we can visualize it and interpret it. So how our single cell RNA sequencing works is we have a gel bead which contains barcodes which will then attach to RNA molecules from the cell, and since the gel bead and the cell are in a water thing and they have to move into an oil, they form droplets because water and oil will mix. And after that we get our data, which then we process, and that my teammate will explain further.
Okay, so now I'm going to move to the first part of cleaning up our data. So it's actually like pre-processing our data. And what we actually do first is QC, or quality control. So basically we throw out bad quality cells. So what do we define as bad quality cells? We kind of have 3 things that we look at. So first is amount of RNA in a cell. So you can see it here, N count is actually the amount of RNA in a cell. And we don't really want either a very big amount of RNA in a cell or not enough big amount of RNA in a cell. So if I were to here on this graph, if I were just to like draw arbitrary lines that I think that represent the data I have to throw away because it's too much RNA or too little RNA, I would do something like this. The second thing that we look at is actually the number of detected genes. We actually look, that is on the y-axis, there. We don't want a very low number of genes because we can't really do a lot of that data. And here, if I were to draw something, I would put it like right there, and I would throw away this data. And the third thing we actually look at is the fraction of mitochondrial RNA compared to the RNA of a cell. And this fraction is important because if you think about it, if we have a bigger ratio of mitochondrial RNA compared to the cell's RNA, it means that the cell membrane broke and that the RNA leaked out. Why? Because mitochondria have, a double layer of membranes, and if something happened to the cell, the RNA in the mitochondria actually was protected and it stayed there. So if this ratio is too big, if we have too much mitochondrial RNA, it actually means that our cell got broken and that is not useful. Here you can see actually the mitochondrial fraction, so you can see that the data with the cells with the biggest mitochondrial fraction, so kind of the broken ones, are yellow, so we also cancel those out. So now I did it kind of like how I was looking at it, but that doesn't really mean a lot because it's just my opinion. So actually we decided to test 4 ways of doing this. One is manual for all samples. Actually, this is the overview, and I'm actually going to talk about each one right now. So first is manual for all samples. So here samples, right? Here's the data, like kind of crumpled up of all of these 6 data. And here we did it manually. How did we do it? We actually looked at the distribution. We kind of zoomed in on the distribution. You can ask me a bit more later if you want. And we canceled out the data that we saw is not well distributed. This is how it looked like for one sample. And then the other method we did was do it automatically for all samples. We actually did this by mean absolute deviation. There's a formula for it, and I can explain that also in depth later. But what is important here is what you can see the difference between the thresholds on the right and on the left. You can actually see that the automatic way is kind of more permissive. It means that it threw away less cells. It threw away just this amount of cells, and our manual threw more. This is kind of a negative side for this automatic way of thresholding because it means that we could potentially keep bad cells and cells that are not useful. The other 2 ways were manual per sample and automatic per sample. So since we had 6 samples, we didn't just see the graph that had crumpled up information of all 6, but we were looking at each sample. So for example, I here show you 2 samples. So per sample manually, each person was doing 1 sample. So we were doing the same thing, looking at the distribution, but the problem with this is it's kind of too subjective. So you can see that one person kind of canceled out this amount of data and another one this amount. So this is a problem because not everyone has kind of the same concept of where to draw a line for the threshold. And automatic per sample, we actually did the same thing, like the same function that we did, but you can see that it's not restrictive at all. So here, almost no cells were cut out, and here a really small amount. So actually, when you sum all of this up, we made kind of a table, and we were looking at what would be the best way to actually do this. So as you can see here, if you like look at it at first, you would maybe think that automated per sample would be the best way. But in this column of the table, which is called filtering low quality cells, it has a minus, which means that actually the only one that had a plus was manual for all samples. It showed that this was actually our winner because it did the most important thing. It basically threw away cells that we don't need, and it wasn't too permissive and it wasn't too restrictive, as you can see here. And then the other part of QC is actually doublet detection. So what are doublets? When our first speaker explained to you how actually sequencing works, you can see that sometimes, so the cells go into the droplets, and sometimes they don't, and sometimes 2 cells can get into 1 droplet. So basically, doublets are droplets containing 2 cells, and this is obviously not something we want. So how do we actually know that we have a doublet? So first, what we do is we simulate doublets from original data, and the way we do this is we take two cells and all the information we have on these 2 cells, and we sum it up and we divide it by 2, and we actually get something called artificial doublet, which you can see in red here. We put them also onto a graph like this, and we can actually see where they are. So here we have the artificial doublets in red, and actually normal cells that are real in other colors. And as you can see, we can already see that these cells have most neighbors that are artificial doublets, and then we can obviously conclude that these are also doublets because they share the same properties. On this graph, you can see the doublet score, which is actually the amount of artificial doublets a cell has. So we can see that the line is drawn here, and we know that these cells are doublets. These are the cells that are here. And this is how we determine that. Now I will give my word to
So now that we've cleaned up our data, we need to sort of pre-process it, crunch it up a bit, because it's insane how much data is generated from single-cell sequencing. So what we do first, before we even start crunching the data, we need to make the cells comparable to each other, which we do through normalization. What normalization is? What normalization is basically you take the RNA count for each gene and divide it by the RNA count for the whole cell. And this basically means that all the RNA counts in a cell will add up to 1, and thus it makes the cells comparable with each other through, in our data. And we multiply this by 10,000 because obviously it's easier to work with. And the reason we need this is because cells don't have the same size, and bigger cells usually have more RNA in them. And we need to compensate for that. And also sequencing is random sometimes, or has random. But now that we've normalized our data, we continue with sort of crunching down our data. And the first step is feature selection and gene reduction. And what this means is that there are roughly 33,000 genes in each cell, which is too much for us to handle, and most of it is not necessary for us to draw our conclusions. So we want to throw out the ones that aren't well represented, not expressed in many cells, so we threw out everything that was in less than 20 cells. We also want to only consider the genes that have a high enough variance to be meaningful in our calculations, and we do this through feature selection, which is a function that shows us, that sets a threshold so we can throw out the genes that aren't variant enough in our data, and this left us with 2,947 genes in the end. But we wanted to go further with reducing our data, and this we did with principal component analysis, which is basically, we have our roughly 3,000 genes, but we want to make 30 principal components out of them, because it's much easier to handle 30 data points. And what we do is we look at genes and try to find similar ones that are expressed in the cells similarly, and just sort of put them together into one data. Do this many times, and in the end, we're left with a few principal components, 30, 40, which describe the whole variance of our data. So now no information is lost, but it's much easier to work with. And in the end, with a second step of this PCA, we reduce it to this UMAP that allows us to represent our data in 2 dimensions and sort of see clusters already. Before we can start actually doing the clustering, we have another step that we must do, which is batch correction. Since our data is generated in multiple steps, we have multiple samples, we must somehow correct for the technical issues that might arise, different temperatures, different people taking the test, things like that. We look for similarities between the samples, correct them, and in the end, we have a graph that is much more together. And once we did batch correction, we go into clustering, which is an algorithm that uses graph theory to build clusters from our data, find similar cells, cluster them together, and name them as probably of the same cell type. We did this, there are different sort of resolutions for this, and in the end we ended up with these 13 clusters, you can see. But now I pass on my hat to Masha.
Okay. So now our next move is biomedical analysis, where we basically give all of this kind of like biological meaning. First thing we need to do is cell-type annotation. For that we need marker genes. They're really important because they're actually highly expressed genes in one cell. So we have our table here, which we have like markers and cell types that expresses these genes. So in this one, in this table, in this plot, actually here, we have like this is CD3E. That's some gene. For example, we did this with like all of these genes. And now this yellow part, that means that in that cluster, that gene is highly expressed. Okay, also we have this plot. Numbers are like clusters, and these are genes. So basically, this is like distribution of genes in clusters. And also like these big circles, that means that all of the analysis. Now we kind of like matched cell types to our clusters. And also there is like compositional analysis. We can see like distribution of cell types in control samples and also in COVID samples. And basically the most important thing here is that in COVID samples we have more like monocytes. We have CD14 plus monocyte, inflammatory monocyte, and also monocytes. So this is like this red part here. So monocytes, most important. And now, here we did Wilcoxon test, which is really similar to t-test. But in this kind of test, we don't assume that data is equally distributed. So here we have like genes in control samples and also COVID samples. And then we can see that like it's not the same. Like not the same genes are equally expressed in COVID and in control. And our last step is pathway analysis. And that basically means that we are checking out if there is like a connection in like which genes are expressed in monocytes in COVID samples, and then how is that connected to some cellular pathways. And now we can see that we have defense response to virus, and also, for example, we have type 1 interferon signaling pattern. So here are results. We can see that COVID patients have like more different type of cells, like monocytes, and healthy have more T cells, CD8 plus T cells, and also CD4 T cells and NK cells. Also, they're different in their genes expressions, and in the end, our signal that these are like diseased cells is that we can see defense response to virus and also type 1 interferon signaling pathways. So, in conclusion,
we concluded that manual quality controls should be considered still in single data cell analysis. Then we concluded that
monocytes and B cells are enriched in COVID PBMC samples, and
we also found out that in COVID, these pathways of viral defense mechanisms and interferon 1 signaling is upregulated. Also, macrophages are also more abundant in COVID cells. And with these conclusions, we want to thank you for your attention. Our project leader from this branch.
Thank you for being such an amazing group.
10 minutes, questions.
I'm interested in how similar do the genomes between the examined cells actually need to be in order for them to be clustered together.
The genomes are the same.
Yeah. What expression, like, what actually makes them able to be put in the same cluster?
So how clustering basically works is you set a resolution which tells the program how many clusters should be expected. But what it basically does is it makes a k nearest k neighbor graph. So for each cell, it builds a graph out of all the cells, tries to find the k and nearest neighbors, and based on this graph, it starts calculating and readjusting its variables to find clusters that have the most connections from this k-neighbor graph, but have the least amount of connection with other things it considers clusters. So it's an iterative method for finding the best thing, the thing that best suits the k-neighbor graph as a resolution.
So how do you make sure, actually how do you distinguish between doublets and cells that are simply in mitosis that already doubles their DNA? And how do you make sure you don't cut out those cells?
Well, there is always a bit of room for error. But actually, maybe there is a technique to do that, but I think here we have so much data that maybe some cells that are like that would not significantly impact. But I think that's a great thing for future research to make it like also inclusive for that type of thing.
Ready? When you stated that the manual method is too subjective and the automatic method is too restrictive, what is the actual principle in the end? Is it a biologically set threshold or threshold setting method that actually defined one you chose to be the best fitting one?
So
Could you
actually,
maybe let Ivana answer?
yes, of course. Yeah.
Yes. So when we do it manually, so basically each of us like got one sample to work with, or two samples, and we decided like subjectively where we put like the thresholds for these lines, which then cancel out the cells behind them, and we decided to go with the manual for all samples because that one like kind of was decided of that one because like it's like canceled the cells that we know are not important, but it also kept some of the cells down here that we still wanted to check out.
If I can just, did this answer your question?
Not really.
Yeah, what it was.
What I was asking is how is this method that you considered a good method defined? Why is this actually?
Oh, so basically we were just, literally there isn't a definition of what a good method is. We were basically just, that's why we wanted to see each of these and literally visually see what happens. This was made by like kind of zooming in and watching the distribution of each of these factors and kind of seeing where it doesn't make sense anymore and cutting out these cells that just look off. And then actually we finished off with this. And then, I mean, we got a result like this from each of these methods. And then when we actually saw it, we compared it and we saw which one actually visually makes more sense. Because you can see like on the graph which cells are kind of outliers. And literally just, it's kind of arbitrary. Yes?
So you relied on human intuition and you stated that it was actually viable.
Yeah.
Cool.
What is the method like compared to actual research on this topic, which is used in actual research?
Can you
Well,
maybe let
well,
Masha say a bit?
Yes. In this.
Can you please repeat the question?
Yes, so the method that you choose in the end for creating thresholds, how does this method compare to the methods actually used in research?
So in researchers, they use like manual for all samples or auto for all samples because the other ones when you do like manual for just one sample and when you do them like one by one, that actually takes a lot of time. So these are like much more efficient.
But yeah, they can use different. It's not that everyone just uses one.
Yeah, they also like choose.
Yeah.
Actually, the table that you have with the
Next
kind
slide.
of, with the summary of the thing. So basically, yeah, maybe if you think a little bit about, or if you explain kind of the column names of the three, that gives a bit more of an idea of why we chose one method over the other.
Should I explain it? Okay. So basically, we have three columns. One is filtering low quality cells. I explained this. This is actually why we even do this. So it's basically how well does it filter them? Is it too permissive? Is it too restrictive? Does it cut off too many cells or not enough cells? Maybe it does nothing. So that's a very important factor. Then we have sample variability. So basically, if we do for all of the samples combined, we don't really take into account that we have different samples. So that is like a minus for when we do it for all samples. And the third thing is objectivity, which is also important because like if we do it like manually by human intuition, that is obviously not objective. Like when we do it with a computer with function and so on. And the function we did was actually I explain later, but yeah, we can explain it too.
Can I just add that with manual thresholding, you look at the distribution of the number of counts between your sample and try to look at the distribution curve, and anything that looks like an outlier, because there will be a bit larger peak at the very end that are the dead cells sort of. There will be a sort of larger end towards the doublet. So anything you see that looks like an outlier and that is outside of this curve, you sort of cut out. And another problem with the automated method here was that our samples weren't since the method uses distribution as well. Our samples, I think, weren't big enough to be properly distributed, and that's why. Stopping from saying something stupid. Our samples weren't big enough by themselves to be properly distributed, even after log transform.
So in pathway analysis, you report adjusted p-value. Can you actually explain why is it adjusted p-value?
This question could be for Ivan as well.
For this one.
Could you please repeat?
Yeah, so you have in pathway analysis.
Just a second. Oh, no. Yes.
Yeah, so you have adjusted p-value. So I was wondering why you don't have just p-value. Why did you have to adjust it, and what did you adjust it for?
Well, I think it's because so we can work with nice numbers. So I'm not really sure.
Yeah, also, Bendy was working on this part, so I know.
I have to admit, I'm not very sure what the adjusted p-value exactly stands for, but I know that this data is acquired when compared to this geobiological process database, and I would think that this adjusted p-value is the p-value acquired from the previous step that was adjusted based on the database. No?
It has to do.
It is a guess.
Yeah, because this was kind of,
It was the last.
this was the last and it has to do with multiple testing. So we do test for many different pathways, and whenever you do multiple tests, you need to kind of consider that in your p-value, and that's what the adjusted p-value comes from. It's kind of corrected for multiple tests. But,
That's
Thank you. Thank you.
Wait.
What's the question? Just a reminder from our previous presentation. So we had two hypotheses for this experiment. The first one was that there will be a significant difference in attitude on e-learning between the control group and the experimental group. And the second was that the personality traits of participants will significantly correlate with the attitude on e-learning of participants. So, what were our methods? So first, one part of the experiment was conducted here at the Summer School of Science in Požega. Here, you were under standardized school test conditions, and so this means you were seated at your desk, you only had a pen, and you were supposed to be like pretty concentrated during the test. And to make the sample bigger, we also made a Google form, so other participants could other students could participate and fill it out online. So first, we had an experimental and a control group. The control group got a text on romanticism, which doesn't really have anything to do with e-learning, so we evaluated that it's a pretty neutral text and won't affect your attitude. And the experimental group got a text from a paper, but we only used the parts that were focused on the negative aspects of e-learning. Yes. Yes. And then you were given a survey. So in the survey, first you had a questionnaire that had like 12 items. So to determine your attitude on e-learning. These items were actually extracted from a different research paper with a similar topic, but the original one had 22 items. The reason we picked this 12, it was actually based on relevance. So in the other research paper, they were also discussing the habits, and for this research, we really wanted to focus on the attitude. And the last part was the IPIP 50 personality test to test your personality. This test uses the Big Five model because it is one of the most used and tested ways. Oh, and actually, one more thing I would like to add. So you may have noticed that the Google form we did online is in Croatian, while you got the one in English. Why? So this is something we took our time to discuss. But since the sample we chose was convenient, which means that the participants were people we know we have like contact to. These are either Croatians or Serbians. And on the one hand, we wanted the conditions to be as similar as possible, but on the other hand, we also wanted the participants to understand the text and the question as well as possible. So in the end, we concluded that this should be our priority, that you understand. And this was also based on the fact that many of you in the school actually asked a lot of questions about certain words in the text. So yes, this is why it is in Croatian and not English.
So our sample consisted of 180 participants, 20 of which were participants of this ST++ camp, and others were given the online survey to upfill. Out of all participants, 62 or 53% were in the experimental group, and 56 or 47% were in the control group. 64% of our participants were female, and 33% male, and 3% others. And the age of all participants ranged from 16 to 21 years old. So after all the data collecting, transcripting, and data analysis, we got our results. So first of all, we did test personality, for which we used Komogorov-Smirnov and Shapiro-Wilk test, and they both showed significance below 0.05, which means we had to reject the null hypothesis, and null hypothesis states that the distribution is normal. So regarding that, with distribution not being normal, we couldn't do the t-test as we planned. And because of that, we did the Mann-Whitney test, which showed us that significance was 0.215, and that is higher than 0.05, which means there is no statistically significant difference between the groups. And we can also see that the mean of control group attitude is 35, and mean of experimental group attitude is 36.5, which is not that big of a difference.
So next we looked at the correlation, and it showed us that only extroversion was significant enough to consider at 0.180, and since it spans from -1 to 1, so you can clearly see it is a really small number, and even though it's significant, it isn't highly significant. Next was model summary. It showed us the percentage of the variance explained by the model. Our model was the personality test, and our percentage was 5.7%, which is also quite low, and showed us it wasn't explained.
So as Lana said, we looked at the correlation between personality traits, their extroversion, agreeableness, conscientiousness, neuroticism, and openness, and attitude towards e-learning. So what's important is we use regression coefficient, or actually like regression method, and it's not necessarily used only for psychology. So regression method is the method that's used in other sciences. So for example, if you want to do some statistics in like math or something else, you can also use this method. And it's actually like consists of two things. One is dependent variable. In our case, the dependent variable was attitude, and it consists out of independent variables. There can be like 2 or more of them. And in our case, those were actually the personality traits. So these. And what the regression method does is it like calculates everything, and then we get the beta coefficient. So that's what we got, and you can see that, as Lana said, the only one that was kind of significant was extroversion. So now to discuss that a little bit, we said that there's no significant influence of personality towards attitude except for the extroversion. We kind of did a little research and maybe like a little thinking, why is that? And we suppose that the extroversion was personality traits that affected people's attitude, because extroverted people are like social and they like to be in groups, they like to study in groups, that's what motivates them. So when we talk about e-learning, there's a lot of group works, there's a lot of like connecting and being online together, and you can kind of reach out for help anytime and talk to people. So that's maybe why extroverts, or actually like people who have extroversion as a personality trait kind of noticed, were the ones who had more positive attitude. And yeah, there's no significant difference between experimental and control group. And why is that? Anza will tell you.
So our experiment had some problems. The main ones were bad sample. So like we said, we only had 118 participants, which is so small that we can't really generalize our results to the whole population. And also our sample was too convenient and non-representable. So our participants were you, participants of the Summer School of Science, and high school students, also our friends who go to high school with us. So our participants are more used to using technology than the average person, and they are more likely to be to use technology in learning. And the second main problem is difference in conditions. So part of the experiment was held here in person, where you were watched while answering questions, while the bigger part of the experiment was held online. So we couldn't really check if the participants were actually reading the tests, which they probably, at least some, haven't actually read it, and if they were answering questions honestly. And so our results could have been incorrect just because people didn't want to spend so much time on, for them, just a form, just serving. Also, the third problem was using an online form to test attitude of e-learning. So everyone who participated in this experiment is more used to using technology than the average person, and the bigger part of the experiment was held online on technology devices. So participants are more likely to be open-minded to the idea of e-learning. So in conclusion, our results showed us that there is no significant difference in attitudes on e-learning between control and experimental groups, and that personality traits, except extroversion, but not significant, don't significantly correlate with the attitude on e-learning. But like I said, there are some problems with our participants, with our sample. So this experiment should be repeated, but without these mistakes. No mistake we did with what we had, but it should be done with more participants. But also it should be done experiment on other persuasive techniques like persuasive images and other.
And for the end, we have a little extra part, which wasn't part of our project, but we wanted to do it because we had data that you gave us, so we ignored the online Google Forms for this part. We just collected your data, and we we calculated the average participant. So this is you. The actual version was 2.98, which is kind of towards the more extrovert, but not really. And you can see the openness was the one that was mostly noticed. It was the most significant one, which means that you all are pretty open to learning, pretty open to new ideas, and all, which is great. And we see the neuroticism, which is also more towards 5 than 1, but that is normal because we're teenagers and also we're not pretty stable. And yeah, that's it. That's it pretty much.
Thank you very much.
Okay, so I have a question about the questionnaire. I saw that there were some questions that are kind of like the same question, but they're just formulated differently. Why did you do that? Is it
So that's used a lot in psychology tests actually, and it's because this way we can see if you're answering honestly and if you're paying attention. So what they usually do in psychology tests, you will have multiple questions that have yes, so they're rephrased just, or you can even put questions like circle 5. So this way you can see if the participants are really paying attention, and if these answers don't match or add up, then you disregard the test.
So you use multiple questions to essentially get the results for a same variable. That's why you use different approaches to get the same variable, right? That's so cool.
Practically, we use the multiply validated IP50, and we just used it. We didn't put it together.
I have a question. Yeah. So what do you think is the actual
actual advantage of using a neutral text for the control group compared to using no text at all? Wouldn't using no text at all for them to have to read produce in the least possible attitude change that you could get?
Yeah, so since we put the article on the experimental group, we needed to do the same for romanticism, and I agree that possibly not using the text would be a good idea, but reading it could maybe make your brain processes differently. I'm not too sure, to be honest.
I'd say that this way the conditions are more similar because if you take your time, like what, 5 minutes to read the text, you're kind of more invested in the experiment. So, like if we were to just give no text and give the questions immediately, maybe you would take like it's just not the same conditions. You would probably answer differently because of something else than the.
If we created a distraction on the experimental group, we needed to make the same kind of distraction.
And it's pretty suitable when you want to like question a few people, but they're in different groups. So for example, our last group were organizers and two project leaders, and we had to have the same amount of people in experimental and same amount of people in the control group. We had to put two of them in experimental and two of them in control group. So by giving them both the text, I mean your argument is completely valid. That's what I thought also. But by doing that, they think they're doing the same task, so they're not like overthinking it. And they're like, oh, he's reading the text too, so it's probably the same, and they don't notice the difference. So it puts every participant through a process of reading the text. Yeah, pretty much.
I didn't quite understand. So you think that extroverts like e-learning more because they can have more like interaction? Because I think, why isn't it the complete opposite, that they actually get more like in-human interaction when they are actually at school, and when they are actually in an environment with other people, when they can like see them and feel them and all of those things that kind of are more suitable for extroverted people? Because I've read so many things that say that actually kind of COVID was good for these people that are more introverted and maybe feel better in their own environment at home. Does anyone else want to?
Well, your arguments are pretty valid, but like I said, our sample wasn't the best, so maybe it was the problem of the sample that just didn't give us the best, the most logical results.
And you also have to keep, you have to keep in mind that the correlation we got is really small still, even if it's significant. So yes, the sample probably is the problem because you can't generalize the population with it.
Thank you.
Do you think the results would have been different if you conducted this test before the pandemic?
Yes, I think it would be different because the only way for extroverts to communicate with people in pandemic was online, and before that it was just casual socializing in person. So it's affected the way people see e-learning and just meeting online.
How do you think the topic choice and how much participants are actually familiar with the topic and have a strong opinion on the topic actually affects how much the opinion will change? So if you had given them a topic which they don't have such a strong opinion for already, would this allow for their opinion to be changed more? Yeah, good question. We talked about that like in our briefing. I think no, actually we didn't. but it's in our final text, so that's why it stayed in my mind. There's a thing called attitude strength. So it's like one of the things that's measured when you kind of actually observe when you look at the attitude. So attitude strength is something that really matters, and it's kind of how much people is prone to changing their attitude. The question is good because, you know, as you said, a lot of people do have a strong opinion on e-learning since they've experienced it. So yeah, probably if they haven't experienced a topic that they were questioned about, the strength of their attitude would be probably lower, so they would be more prone to changing their attitude.
There was one research when Pluto, when they removed Pluto from the planet, when they announced that Pluto isn't a planet anymore, they conducted a research on, I think, 6th graders, and they were persuaded into thinking that Pluto isn't a planet anymore, and as we know, 6th graders don't have a lot of knowledge in astronomy, and then they were questioned about what arguments could they give that Pluto is a planet and that Pluto isn't a planet. So yeah, that's an interesting fact.
A question maybe for Angela first. You mentioned that you had a control test which was on romanticism. So personally for me that text wasn't very neutral because it actually spoke a lot about, I mean it was anti-science, like it spoke how romanticism was in science and how they wish to go back to, how they idealized the medieval period. Basically how it affected me was that I kind of then was really like into computers and e-learning because I got really annoyed by idealizing the medieval times. So my question is, do you think that the text was really neutral? And could that maybe affect the results?
I haven't actually read the text, so I don't know how persuasive it is. But it should be pretty neutral, and our goal for the control group was not to give any opinion, not to put opinion into your head, and to be pretty neutral.
So you're basically saying compared to your other non-controlled tests, even if it wouldn't be very neutral on its own, but compared to the other text, it's much more neutral.